you're only going to be responsible for the information that we're about to go over, and you're only responsible for it in lab. You will not be tested on chapter 4 in lecture, only in lab.
The stuff that you got, and you going, is it going right? I'm printing it off, and it's going right with it.
That's
Okay.
the stuff we're talking about. Yep, you're only responsible for what we go over.
And this stuff, the objectives that you're talking about, they in lab, the lab part of it.
Right, they're in the lab, Blackboard site. Yep.
Here it is.
All right, everybody ready?
See?
I printed it off already because I know it.
All right, so chapter 4, the reason we're doing it in lab is because it covers material that happens in lab. And we're talking about essentially what's going on in that incubator when we leave. Okay. So starting off, the very first part of chapter 4 talks about bacterial growth. Growth for humans we think of as getting bigger, right? We grow up, we get taller, we get bigger. Bacteria growth is not an increase in size like it is for humans. It's an increase in number. So bacterial growth is an increase in number, not an increase in size. Bacteria grow by going through a process called binary fission. It's an increase in number, not an increase in size, and they grow using a process called binary fission. Binary fission is how they reproduce, it's how they grow. So we start off here with one bacterial cell, and basically it divides in half. It becomes two bacterial cells. So growth, increase in number. We had one, now we've got two. Then those two will go through binary fission and become 4. Right? And those 4 will go through binary fission and become 8. So every time a bacteria goes through binary fission, the total number of them doubles. So then that gives us this term doubling time. The amount of time it takes for bacteria to go through binary fission is the doubling time. How long it takes to go through binary fission. And it varies. It's not the same for every bacteria. Different bacteria take different amounts of time to go through binary fission. And it also depends on conditions. Do they have enough food, enough water, the right temperature? Right? If it's too cold, too hot, it's going to slow it down. Okay. If they don't have enough food or not enough water, that's going to slow it down. So under ideal conditions, assuming they've got the right temperature, the right amount of water, everything they need, this can be as little as 20 minutes. Doubling time can be as little as 20 minutes under the right conditions. So, let's say you're at home making potato salad, right, for a picnic or something. Nothing you've got is sterile, right? You're not autoclaving your bowls and spoons at home, right? So everything that you're using has bacteria on it, and that's okay, right? We're used to that our whole lives. Everything that we've eaten has been made in bowls that have bacteria in it. So let's say you make your batch of potato salad and just throw out a number. Let's say there's 10,000 bacteria in the whole bowl. There's probably more than that, but let's just go with 10,000, okay? You leave that potato salad out at the picnic, 20 minutes go by, you've got 20,000, right? 20 more minutes go by, you've got 40,000. That's why you've got that 2-hour time limit, right? How long things can sit outside of the fridge before they have to go back in. And that's why refrigeration helps preserve food, because the temperature is cold. Most bacteria either can't go through binary fission or it's slowed down significantly in the refrigerator. That's why we move all of your stuff to the fridge between Wednesday and Monday, so that it's still alive. The growth is so slow in that cold temperature that they live longer. Most food spoilage is done by bacteria. Refrigeration slows down their growth so the food lasts longer. That makes sense?
Question. I want to know how bacteria it could play a role in this. I had gotten food poisoning with tuna fish, Mason tuna fish, many moons ago. Left it in a can, put it in a refrigerator, and ate it the next day. So is that a bacterial thing, or?
Probably. It probably wasn't anything to do with the metal. It's probably bacteria.
Okay.
That's just guess.
Okay.
Yeah. Okay, so we're good with binary fission and doubling time. Okay. Factors that affect the growth of bacteria. Remembering that growth means binary fission. Okay, so factors that affect binary fission, how fast they go through it. We've already alluded to the first one, right? Temperature is a big factor that affects how quickly they grow. Too cold slows them way down. Too hot will also slow them down, right? Possibly kill them if you get it hot enough. So temperature is a factor. We've got 3 different groups of bacteria here based on which temperature they prefer. Not all bacteria like 37 degrees, right? That's not the best temperature for everybody. That's just the best temperature for the bacteria that we work with. Okay, so don't assume that every bacteria likes 37 degrees. for the mesophiles, that's where our bacteria are. They grow best between 25 and 45 degrees Celsius. So they go through binary fission, they grow best between 25 and 45 degrees Celsius. They prefer 37 degrees, right? So they'll grow between 25 and 45, but they prefer 37. So that's what most of our bacteria are. They're mesophiles, 25 to 45. Psychotrophs, they prefer 20 to 30 degrees Celsius. They grow best between 20 and 30 degrees Celsius. So if we were trying to grow those, we'd turn the incubator down. And they're most of the bacteria that do eventually spoil food, even in the refrigerator. It's these guys, because they can grow in cooler temperatures.
What do you say the temperature?
20 to 30 degrees Celsius for psychotrophs. That's why you can't leave stuff in there for more than a week or so before you have to throw it out. If it's something that you made fresh and it's not full of preservatives from the manufacturer. Okay. Jelly you can keep in there for a while because they loaded that stuff up with preservatives. But if you made something yourself, then you've got a week, maybe two at most, that it can stay in there depending on what it is. Thermophiles are the opposite end. They prefer the warmer temperatures. They grow between 45 and 75 degrees Celsius. 45 to 75. So again, if we were growing any of them, we'd turn off the incubator. They wouldn't grow very well at 37.
Do they have temperature?
No, there's not one specific one. It varies by organism.
What's the name of that one?
Some of the ones that live in your hot springs, like those domain archies that we talked about, those extreme environments, a lot of those are thermophiles. All right, so we go with temperature. Second major factor, oxygen requirements. Not all bacteria like oxygen, and some of them prefer different amounts of it. Obligate aerobes. If you're obligated to do something, what does that mean?
You have to.
You have to. You are all obligated to take your test, right?
I heard that name before.
Obligate aerobe. What's aerobe mean? Aerobic.
Terrible.
With
Air.
oxygen,
With
right?
oxygen.
With oxygen. So these organisms have an absolute need for oxygen. They have to have it or they will die. Obligate aerobes. Absolute need for oxygen. They die without it.
Oh, you ain't gonna fill it in.
Yeah.
I was wondering, like, how do you make that?
That's what I'm telling you. The term is obligate aerobe. The definition is the organism has an absolute need for oxygen. So we got no problem growing those. Plenty of oxygen in the incubator. Right? Those are nice and easy. Second category of bacteria, obligate anaerobes. Anaerobic is without oxygen. These guys are obligated to not have oxygen. Right? So they do not like oxygen at all, and oftentimes oxygen will kill them. So they grow in no oxygen, zero oxygen environments. Oxygen often will kill them. So are they going to grow in our incubators? No, tons of oxygen in there. You have to have special carbon dioxide incubators. CO2 gas that pumps in every time you open the door, the tank comes on, the CO2 gas fills the chamber, pushes the air out. You've got to work in special conditions for those. Third possibility there, facultative anaerobes. Facultative means flexible. These bacteria are flexible. They can grow with or without oxygen. They can grow with or without oxygen, but they grow better with it. So facultative means flexible. They can grow with or without oxygen, but they grow better with oxygen.
So profound.
Yep. That's different than the obligate aerobes. They can't grow if there's no oxygen around. Right? These guys can, but they like it better if oxygen's there. Microaerophiles. Micro means?
Small.
They prefer small amounts of oxygen. Prefer small amounts of oxygen. So to grow these is pretty interesting. You can work with them out in the room, regular oxygen-filled environment. Do all of your aseptic technique. And you got to get yourself a big jar, like one of those big, tall pickle jars. The big pickles come in, screw cap on it. So what you're gonna do is stack up your Petri dishes in the big jar, the big pickle jar. It's actually called a candle jar. You can buy official ones, or you can just use the old pickle jar. Okay? And then you stick a candle in with it. And light the candle. It's my best drawing ever, guys. I have candles. Okay? Be proud. Okay, so you light the candle, you've got your Petri dishes inside, screw the lid on the pickle jar or your candle jar, and then put that whole thing in the incubator right at the right temperature. What will happen since you've screwed the lid on is the flame will use the oxygen that's in the container until there isn't enough left to support the flame, right, and then the flame will go out. There'll be only a small amount of oxygen left, the microaerophile environment. So they need small amounts of oxygen. Too much might kill them.
What's that called?
It's a candle jar. It's okay. It's a candle jar.
We need to know
And
that.
again, yeah. Why would I tell you if you didn't even know that? You light the candle, the flame burns up most of the oxygen in the jar, so you're left with a microaerophilic environment, only a small amount of oxygen left. And again, you just stick that whole jar in the incubator at whatever temperature that bacteria prefers. And this last one down here has two names, aerotolerant anaerobes, also known as obligate fermenters. So two terms for the same group of organisms. Aerotolerant anaerobes. Aerotolerant anaerobes, let's start with that one. Aerotolerant, they're tolerant of oxygen, right? They don't care if oxygen's present or not, right? It's got anaerobic here, so without oxygen. They can grow with or without it, and they have no preference. They'll grow just as well with oxygen as around as they will without it. So they don't care. They grow just as well with or without oxygen.
No preference.
No preference. They're also called obligate fermenters because they do fermentation. That's why they don't care if oxygen's there or not. Fermentation will happen without oxygen, with it there, it doesn't matter.
Say that last part one more time.
They're obligate fermenters because they do fermentation, and fermentation happens with or without oxygen. All of these other guys don't do fermentation. They use other types of metabolism that requires oxygen or something similar. These guys do fermentation only. No oxygen is needed for them. But oxygen won't kill them. They'll still grow. So, everybody got those differences? Okay. So you gotta have the right temperature, the right amount of oxygen. The next thing that you're gonna need is the right pH. Okay, so remember from lecture, buffers prevent changes in pH. All of the media that you guys make on the nutrient broth, the nutrient auger, has buffer already in it to keep the pH constant at whatever level that particular mix is set for. Okay, so we've got buffers in there. And almost all the bacteria we work with are neutrophiles. What do you think that means? They prefer neutral pHs, right? So they prefer neutral pHs. They'll grow between 5 and 8, pH range of 5 and 8, but they prefer 7. Okay, so neutrophiles will grow with a pH between 5 and 8. So that's a little acidic to a little basic.
pH, but
Prefer 7. Yep, will grow from 5 to 8, but prefer 7. So before refrigeration, before we invented refrigerators, if you were going to preserve food, a lot of times you would pickle it. Right? With those giardiniera salads, the Italians like them with carrots and cauliflower and broccoli, or just pickles and cucumbers. All of that pickling works because you're getting the pH down below 5. So the bacteria, you may not refrigerate it, but the bacteria don't grow well at that lower pH of the pickle, the brine that their food is in.
So pickling means?
Pickling has acid in it, and the acids in the pickling liquid, in the brine, bring the pH down so that these guys are not going through binary fission. Remember, growth is binary fission. So pickling drops the pH,
So bacteria won't
so bacteria won't grow. That was a good way to preserve food before refrigeration.
Is that the same thing as like canning? Because I know my relatives from West Virginia can
Everything. Yeah, my
and
grandparents
for
did too.
years.
Well, most canned things aren't necessarily acidic. Pickles, the spices that you add do drop pH,